At this point, then, I'd like to go for our final talk of the discussion today, which is from Guy Cochrane, who's the head of the European Nucleotide Archive, which is at EMBL's EBI, or the European Bioinformatics Institute. So Guy, looks good?
Great. Well, hello, everybody. Thanks very much for the introduction, Justin, and thanks also to all of the organizers for the invitation. It's a great pleasure to be able to be part of this series. So the scientists today looking at marine environments, looking at life in the oceans, has at their disposal a huge array of really powerful technologies to understand, to analyze, and to probe. These technologies are high resolution, they're deep, and they are high throughput, they're very broad. So you could consider sequencing that we just heard about, DNA sequencing, where we can very deeply profile organisms and resolve. On the imaging side, we've seen lots of beautiful images already. We have microscopy, we have all sorts of high throughput and high resolution methods available there as well. Of course, for people studying living things within the oceans, it's important to know the abiotic components of the environments in which they live. And of course, in parallel, outside the biology world, there's a whole host of technologies that have really come of age to provide access to environmental data, oceanographic data, hydrology data, and so on. Think of the advances in autonomous sensing, streams of continuous data. Think of the advances in remote sensing. So for the marine scientists, faced with these very complex, very deep, and very broad, very powerful data sets, there are great opportunities. In order to benefit from any of these data sets, the work cannot be done immediately. No one study that generates data really can any longer expect in itself to fully resolve all of the science that will come from the data set. There's simply a lot more value in there. And so what can happen is over time, whole communities of scientists can work on the different data sets to gradually extract the value and learn science, derive knowledge from those data sets. So what's become extremely important is that one takes an approach to these data that is highly organized and structured and appropriately shares data, preserves data, and makes data available and reusable by whole scientific communities. So what I want to do is focus on the data. I'll start first by introducing Tara Oceans, a project that's been mentioned already. This is an initiative in life science investigation of the oceans that I've been fortunate enough to be involved in, or to get involved in a few years ago. I'll talk a little bit about the approach that Tara Oceans has, and I'll talk about the data that it produces, and then I'll switch over to talk a bit more generally about the journey that these scientific data take through what is a broad, global, open data infrastructure for adding value and making these data accessible and usable. And then I will cover such items as data standards and access to the benefits that arise from the data and the science they represent. And then I want to flip back to the Tara initiative at the very end to highlight some of the impacts, some of the outputs that Tara has been able to achieve by taking this structured approach to the data. So this is the Tara, the vessel. She is a vessel that's been in scientific research for several decades. In the last 10 years or so, that's been dedicated to the study of living things. And on the right here, you can see some of the, I move that, there's a thing blocking, there we go. You can see some of the missions, typically voyages that Tara has been on. The current mission is Mission Microbiomes. The ship is somewhere, I think, off South America at the moment. When I give some statistics and some examples later on, I'm going to be talking more about a previous exploration, a 3-year global circumnavigation that you see here, where each of these grey areas is an event. So there's a huge number of sample points along the way. So Tara is ambitious. She concerns herself with primarily with the plankton, the planktonic layer, but at many scales of organization. So there is an interest in understanding the evolutionary relationship between organisms, understanding the biochemistry, the way in which, or the function of individual organisms, understanding how one organism interacts with another, and how you build that up into whole communities, and ultimately scaling all the way up to how communities and whole ecosystems contribute and respond to other planetary systems, how they contribute to their health and changes on planet Earth. So here we're looking at the methodology. So Tara goes on a voyage and stops at various sites and takes samples. So the sampling of the water column is at various levels. So it goes down to almost a kilometer using different types of instrumentation. And concurrent with the sampling, there are always measures of various physical and chemical oceanographic parameters, temperature, pH, and so on. So there's a whole host of environmental parameters that are captured with samples. But the samples are preserved and filtered, size filtered and preserved in different ways for later sequencing analysis and imaging analysis. And on the sequencing side, it's a diversity of different sequencing applications. So here we have the isolation of organisms or the amplification of individual organisms and the derivation of genomes from those organisms. Community DNA studies where you take anonymous samples of large numbers of organisms and you mix their DNA together. You have a mixture of DNA. And you can study that using metabarcoding, which is where you identify, you look for occurrences of certain organisms or abundances of certain organisms based on reference sequences, so certain loci that are characteristic of species or strains. Metagenomics is where you reconstruct whole metagenomes from fragmented reads from these anonymous populations of molecules to reconstruct the individual genomes of what was there. Metatranscriptomics on the right here is where you take RNA, you do RNA sequencing, and you start to look at the activity of the different genes that you find in a particular sample. And then from that, you can infer or begin to infer the biochemistry, the function, the activity of that particular community of organisms that you've been looking at. So in parallel on the sequencing side, there are a whole host of different applications at different scales. We have the very high resolution microscopy, flow cytometry, and then slightly lower resolution, but probably higher throughput. We have lots of morphology data available from a host of other methods. So Tara collects many samples, captures environmental data from those samples, captures sequence data, and captures imaging data of all sorts of different types. And just on the bottom right here, we see the kind of organization for the data themselves. There is a sample, there is a registry of samples, there are metadata relating to the sampling process, and then to that we attach the environmental data, the sequence data, the nucleotide data, and the imaging data. And so right from the start, Tara has taken an approach to proactively structure, organize the data, and make the data available as a community resource. So from the start, this has been the data release policy to make data available as soon as possible. So raw data from the images, from the sequencing, and then the early analysis on those data sets. Much of this happens before there's any scientific interpretation, before there's a scientific publication, literature publication. And then there's an ongoing effort to curate the data to make sure that the descriptions of the samples are correct and to enrich the descriptions of the samples with further information that is external. And so one part of organizing data well is to follow data standards, but the kind of interdisciplinary cross-domain data standards that were required, of course, needed to be developed. So the Tara consortium, amongst other groups working on marine sampling, got together back in 2014-15 to really try to pin down a way of describing and structuring a description of marine data sets for the purposes of the kind of study that Tara and other initiatives were taking on. And this is the M2B3 data standard. It really defines, it doesn't define how you do your science, it defines how you describe your science in a way that allows people to discover, to compare, and to contrast, and so on. And really it's an intersect between details of a sampling process, details of that environmental, that contextual information when a sample was taken, but also information about the sort of large observational biodiversity information that you would want to bring in and intersect as well, and various other things. So incidentally, this standard remains in use by the Tara Oceans efforts as they continue, but it's been adopted by other groups, by many other groups, and it's been informative. So people have certainly been in designing ways of handling their data in more recent studies. It's been informative for that process as well. And so Here, just looking at some of the numbers. So this is from back in 2015. So these were the first wave of publications and analyses that came out of the global circumnavigation. So it was a large number of samples overall, 35,000, and thousands of data sets. What was then, this has been very heavily surpassed, but what was then a very large volume of data. And of course, this huge potential to discover many different things. And in the first analysis, looking at the prokaryotic section, there was an identification of there's a creation of a catalog of some 40 million genes that have been identified. So just to show that this really is science, marine science at scale, and a resource that's made available and can and has been mined long since. So this gives me a chance to switch over. As I switch over to the data part of this, I've become a little bit more generic. This is not just about Tara. It's not just about marine data. But this gives me a chance to introduce my part of EMBL. So this is the European Bioinformatics Institute, and Cornelius has already given the introduction to EMBL, so I'm grateful for that. And he mentioned the service function that we have. And what we're seeing here are many of the data resources that make up the informatics, the bioinformatics data services function that we operate from the European Bioinformatics Institute. And in here we have databases, tools, analysis systems, support, training, and so on. And I'm going to point out in particular ENA, which is the European Nucleotide Archive. This is the sequence database. It's the European part of a global database of record for sequencing data. We have a sample database. We have Magnify. I'll mention this in a minute. This is about metagenomics analysis and many other databases. So typically at EBI, we run the European part of global deposition databases, repositories of data, and we provide layers of analysis and service and value add on top of those to make these data really accessible and usable to people. So putting data in a database is really just the first step. So Tara will put her data, here I'm taking the example of sequence data, into the ENA system, and immediately there will be a propagation, a dissemination of those data. The data gain added reach. And so here we're looking. This is just within the one institute, just within EBI. We're looking at the different data resources that we operate. This is the ENA. And every time you have a band here, this represents a data flow between two different data resources, two different databases, or a data flow or a cross-reference or a link of some sort. And so as soon as the data are put into a database like ENA, there's an immediate and fairly autonomous propagation of those data into different databases. So this is a protein database. This gives us information about a particular protein from the Tara data set. These are protein functional domain databases. They give us information about how proteins are structured that come from that Tara set. This is Magnify. This is a set of analyses, visualizations, and tools to interpret metagenomics data, for example, and metabarcoding data, and so on. So as soon as the sequence data appear from Tara, they become available through these systems. And it's fairly autonomous because these are established just to do this. So that's within EBI. When we look outside the Institute, of course, there are many, many more layers of databases. The ENA is part of the European e-infrastructure Elixir, which is the coordinating point for bioinformatics data resources in Europe. It is connected through a partnership with NCBI in the US and DDBJ. And there are many other generalist and different databases that will pick up the data, do some curation, add some value, and make the data available in different ways to new people. And of course, some specialist databases. Here we're looking at the Marine Metagenomics portal from Norway. This is general for marine metagenomics data. Then we have specifically for Tara, a project from France that builds atlases, gene atlases, barcode atlases. These are just representations. There are many of these. It's difficult to estimate, but there are possibly several thousand databases of different types that pick up these data that come from projects like Tara and make them more usable and more available and more accessible to people. So this is a project on the European side. So Europeans will know that the European Open Science Cloud is the major vehicle through which the European Union will provide scientific data infrastructure for its scientific programs. And here we are building in one project the blue cloud. So this is the marine and the other aquatic informatics services to deal with marine science and aquatic science. And so this is bringing in the sequences, it's bringing in images, but it's bringing in all sorts of other marine relevant data types as well, and providing such services as data discovery and access, virtual research environments and catalogs. And this is only really possible because we've had a structured approach to data. And the TARA data will of course be available through here. And so on the right, we're looking at a demonstrator of some of these services that are being built. This is called the Plankton Genomics Demonstrator. And what we're doing here is, to start with, we're looking for environmental correlates. So we're looking at species distributions, and we're looking at the environmental conditions with which those distributions correlate. And then we're taking data from elsewhere in a second step and extrapolating. So sampling, of course, is sparse across the world's oceans. So we're extrapolating using the conditions we know to exist from different models in different parts of the oceans, and coming up with global plankton biogeographies as an extrapolation. So the oceans, we heard from Minhan that the oceans really are important for everybody. These are they provide us with food, they help us combat climate change, they're possibly impacted by climate change, they provide oxygen, they remove carbon, they provide leisure activities, and they support our transport system. So they're really important for everybody. So the science around oceans really has to be global. There are a couple of important discussions going on at the moment, politically, from the Convention on Biological Diversity and from other parts of the UN, around how we handle scientific data that relate to marine environments. So for the CBD, it relates to coastal environments. For the UN process, this is for the high seas outside those areas of national jurisdiction. And really, it's working out how, in a world where you have wealthy countries and countries that have lower incomes and are less able to engage so easily with science, and certainly scientific exploration, which is a costly endeavor, of course, how everyone can benefit from that system. And I believe very strongly that actually the openness of the data, providing global accessibility to the data from all of these studies, is really an important element of allowing the benefits of this work to be shared with everybody. Part of that is the data, part of that is allowing scientists who are contributing to be credited for what they do and to have attribution. And a part of it is really building a globally connected scientific community to work on these data sets. So finally, I look back to the impact so far that TARA Oceans has had. Here we're looking at some of the higher profile publications and special issues from the project. So from the consortium itself, it's a big consortium, but we've had some over 130 publications. And you could do go and have a look in here. There's an awful lot of reading material here to get stuck into, but some very interesting science. But what I want to focus on is really a selection of examples of studies that don't come from the consortium. These studies come from other scientists who have picked up the TARA data through the infrastructure that I've presented, and developed new scientific ideas and explored more. I won't go into huge detail because I'm running out of time. Here we are looking at, for example, discovery of a new group of rhodopsins. Here we're looking at biomass distribution across the oceans and elsewhere. Here we're looking at selection, and in particular how dispersal is restricted by environmental conditions in the plankton. Here we're looking at the diversity of phages across the world's oceans. Here we're looking at the impact of particular codon usage adaptations or codon usage changes in protecting against certain resource-related characteristics of organisms. And here we're looking at using TARA to compare with data from over 100 years ago to look at the calcification, the extent of calcification in foraminifera. So looking at the impact of climate change, anthropogenic climate change on these calcifying plankton. So they're just a selection of the kinds of science that have happened based on the TARA data set so far. So to acknowledge some of the contributors, I can't cover everybody. Many people work on TARA Oceans. Chris Bowler and Stefan Pizon in particular provided some of the slides and some of the materials I've used. the coordination group, the expeditions group, and I mentioned as well Blue Cloud. It's a broad project, but the demonstrator that I mentioned comes from EBI, Sorbonne University, and FLIS in Belgium. And just on the right to mention some other projects, some of which we mentioned already. Atlantico is ongoing and brings together 5 flagship cruises. And of course, we've heard already about the planetary biology program and the ecosystems interest from EMBL. And I thank you for your attention.